Good evening all. Today we are going to discuss about host pathogen interaction immunology, adaptive immunity and antibody. So you all know that antibody is part of adaptive immunity. We have already talked about innate immunity. So where what happens in our body, the immune cells which are part of innate immunity acts upon. But also when we grow up we also face different kinds of substances which are pathogens, and against it we generate adaptive immunity. So antibody is part of adaptive immunity. Now we have already heard that B cells produces antibodies. So an antibody is uh, obviously it's a protein, and it's made up of two chains, heavy chains and light chains. So in the light chain you can see the structure of light chain and heavy chain. So here we in the previous class we already discussed about heavy chain. So today we will discuss about light chain. So when so in the this is heavy chain and this is light chain. So in this heavy chain and light chain you can see this is the antigen binding domain, and in this constant domain it has effector activity. Okay. When amino acid of light chains were sequenced, a pattern emerged. Amino terminal end, that is 110 amino acid, varied. Other part remained constant, were found to be either kappa or lambda. In human, 60% is kappa and 40% is lambda. In mouse, 95% is kappa and 5% is lambda. Heavy chains, amino terminal end also shows variability. 5 different heavy chain constant regions is called isotypes. IgM, also called mu, IgG, also called gamma, IgA, also called alpha, IgD, this delta, and Ig is epsilon. Some subisotypes have been discovered in some species. Each antibody has 2 identical heavy chains and 2 identical light chains. If you have any question, please let me know. So each antibody has 2 identical heavy chains and 2 identical light chains. Chain composition of the 5 immunoglobulin classes in humans. Class IgG, heavy chain is called gamma. Subclasses gamma 1, gamma 2, gamma 3, gamma 4. Light chain kappa or lambda. Molecular formula gamma 2, kappa 2, gamma 2, lambda 2. So IgM, heavy chain mu, subclasses none, light chain kappa or lambda, mu to k2 n, and mu to lambda to n equals to 1 to 5. So it can be pentameric. IgA heavy chain alpha, subclasses alpha 1, alpha 2, light chain kappa or lambda. Molecular formula alpha 2, kappa 2, N, alpha 2, lambda 2, N. IgE epsilon, subclasses none, light chain kappa or lambda, epsilon 2, kappa 2, epsilon 2, lambda 2. IgD delta, subclasses none, light chain kappa or lambda, delta 2, kappa 2. So gamma delta alpha. So here is antigen binding site is there and biological activity is there in the constant region. Hinge region gamma, delta and alpha heavy chains have extended peptide sequence, rich in proline and cysteine, gives flexibility. CH2 and CH3 contains oligosaccharide side chain. Mu and epsilon present in secreted immunoglobulin and membrane bound immunoglobulin. No hinge region is there. So additional domain is there which is CH2. Immunoglobulins can be secreted or membrane bound. Membrane bound differ in their carboxyl terminal end. Extracellular spacer of 26 amino acids is there. Hydrophobic transmembrane sequence and cytoplasmic tail. So this is IgG. This is variable light chain, constant light chain, variable heavy chain, constant heavy chain. This hinge region is present in IgG. In IgD you can see the kind of similar IgE, IgA which can be dimerized. It has J chain also which helps in solubility. IgM can be pentameric and IgM are pentameric. So you can see disulfide bonds are present in between 5 monomers and one G chain is present. IgM pentamer monomeric IgM expressed on B cells and its secreted form is pentameric. First class produced in memory response, activates complement, very good at agglutination, 5 to 10% of total serum immunoglobulin. IgD membrane bound on B cells. IgG most abundant, 4 human subclasses, crosses placenta, involved in complement, 80% of total immunoglobulin. IgG1 disulfide bond is there, IgG2, IgG3, IgG4. IgA, so it's dimer, it has a J chain, predominant class in secretions, exists as dimer, can cross-link large antigens, daily production is maximum among other isotypes 5 gram to 15 gram, present in mucus. Structure of a secretory IgA, J chain. So the J chain it's basically a secretory product, a secretory compound. So it helps in transportation of immunoglobulin A. So what happens after producing from plasma cells dimeric IgA? So it gets bound with a poly Ig receptor and this can engulf in a vesicle, and after enzymatic cleavage, it secretes as a secretory IgA. IgE involved in allergic reactions, involvement in parasitic infections as well. So here the property and activity of different Igs are listed, and also along with their molecular weight. So once again I would like to introduce myself. I'm Ananya. I'm teaching for the course Host Pathogen Interaction. Our supervisor of the course is Professor Himanshu Kumar. If you have any doubt, you can reach to us with any of your doubts. Dilemma. Since only 31 to 35,000 genes in the human genome actually encode proteins, how antibody diversity between 1 million to 100 million specificities achieved with such a limited number of genes? What causes the difference in amino acid sequences? How can different heavy chain constant regions can be associated with the variable regions? Genome in mature B cells is not same as other host cells. So in germline DNA, multiple gene segments code portions of single immunoglobulin heavy or light chain. During B cell maturations and stimulations, gene segments are shuffled, leaving coding sequence for only one functional heavy chain and light chain. Chromosomal DNA in mature B cells is not the same as germline DNA. Key contributor in this field is S. Tonegawa. So, Tonegawa's experiment in immunoglobulin genes rearranged. So, we'll talk about Tonegawa's experiment in immunoglobulin genes rearranged and how this immunoglobulin gene gets rearranged and then forms this antibodies. So, we'll talk about it. Before that, if you have any doubt, let me know. So, there are germline genes where you can see VN, V2, V1, J, C, and so on. And there are different sites of restriction enzyme digestion. Okay, so how this rearrange happens? You can see some portions gets deleted and rearranged and can form this kind of strings. Like VN means any number of V. So V2, V1, and then V1 gets get linked with J, and like that. So there can be different experimental basis for diagnosis of rearrangements at an immunoglobulin locus. This is cell IgG expressed. So this is hematopoietic stem cells, and here you can see from hematopoietic stem cells lymphoid cell is generated, then pro B cell with the help of partial heavy chain gene rearrangement, pro B cell to pre B cell, complete heavy chain gene rearrangement, pre B cell to immature B cell, light chain gene rearrangement, immature to mature B cell. Here change in RNA processing happens. Mature B cell to activated B cell, it stimulates antigen, and then differentiation where IgM secreting plasma cells, and class switching happens where memory B cells and then different plasma cells secreting various isotypes gets developed. So this until this process it happens in bone marrow, and from there it happens in peripheral lymphoid organs. So kappa and lambda light chain segments. L leader peptide guides through ear. V and J and C is a constant region. VJ segment codes for variable region. Heavy chain LVDJC. VDJ segment codes for variable region. So genetic organization of immunoglobulin globulin germline gene segments. Lambda chain DNA is kappa chain DNA, heavy chain DNA. So there are different chains, and here you can see different gene segments, and this gets rearranged and then form the different isotypes or different subtypes. Variable region gene rearrangements. Variable region gene rearrangements occur during B cell maturation in bone marrow. Heavy chain variable region genes rearrange first, then light chain variable region. In the end, B cell contains single functional variable region DNA sequence. Heavy chain rearrangement happens after stimulation of B cell. So this is heavy chain rearrangement. I will discuss the heavy chain and light chain rearrangement in detail in some time. This is light chain rearrangement. Okay. Generation of antibody diversity. Multiple germline gene segments are there. Combinatorial VDJ joining by VDJ recombinase. Recombination activating genes 1 and 2 and terminal deoxynucleotide transferase. Junctional flexibility. P region nucleotide addition via recombinational signal sequence. N region nucleotide addition via recombination signal sequence. Somatic hypermutation. Combinatorial association of light and heavy chains. Ensuring only one chromosome expresses the antibody genes allelic exclusion, that the rearranged heavy and light chain genes from only one chromosome are expressed. Model to account for allelic exclusion. This is junctional flexibility in joining. Class switching. Switch region, switch recombinant, switch factor, activation induced cytidine deaminase. Antibody diversity, multiple gene line segments and combination of those segments. Mechanism of variable region DNA rearrangements. So, as we were discussing about antibodies, so I would like to follow this textbook, Kuvi Immunology, as well. So here you can see this is an example of IgM. So antibodies are the antigen binding proteins, and those are present on the B cell membranes. Antibodies are present on the B cell membrane and secreted by plasma cells. We have discussed this so far. So, membrane bound antibody confers antigenic specificity on B cells. Antigen specific proliferation of B cell clones is elicited by the interaction of membrane antibody with antigen. So, secreted antibodies circulate in the blood where they serve as the effectors of humoral immunity by searching out and neutralizing antigens or marking them for elimination. So all antibodies share structural features, bind to antigen, and participate in a limited number of effector functions. The antibodies produced in response to a particular antigens are heterologous. Most antigens are complex and contain many different antigenic determinants. And the immune system usually responds by producing antibodies to several epitopes on the antigen. This response requires the recruitment of several clones of B cells. Their outputs are monoclonal antibodies, each of which specifically binds to a specific or single antigenic determinant. Together, these monoclonal antibodies make up the polyclonal and heterogeneous serum antibody response to an immunizing antigen. So, we'll discuss about basic structure of antibodies. If you have any doubt, you can let me know. So, basic structure of antibodies. Blood can be separated in a centrifuge into a fluid and a cellular fraction. The fluid fraction is the plasma, and the cellular fraction contains red blood cells, leukocytes, platelets. Plasma contains all of the soluble small molecules and macromolecules of blood, including fibrin and other proteins required for the formation of blood clots. If the blood or plasma is allowed to clot, the fluid phase that remains is called serum. It has been known since the turn of the century that antibodies reside in the serum. The first evidence that antibodies were contained in particular serum protein fractions came from a classic experiment by A. Tisselis and Cubbard. They immunized rabbits with a protein of ovalbumin and then divided the immunized rabbit serum into two aliquots. Electrophoresis of one serum aliquot revealed four peaks corresponding to albumin and the alpha, beta, and gamma globulins. The other serum aliquot was reacted with ovalbumin and the precipitate that formed was removed. The remaining serum proteins which did not react with the antigens were then electrophoresed. A comparison of the electrophoretic profiles of these two serum aliquots revealed that there was a significant drop in the gamma globulin peak in the aliquot that had been reacted with antigen. Thus, the gamma globulin fraction was identified as containing serum antibodies, which are called immunoglobulins. To distinguish them from any other proteins that might be contained in the gamma globulin fraction, the early experiments of Cubbard and Tisselis resolved serum proteins into three major non-albumin peaks: alpha, beta, and gamma. We now know that although immunoglobulin G and the main class of antibody molecules is indeed mostly found in the gamma globulin fraction. Antibodies are heterodimers, so antibody molecules have a common structure of four peptide chains. This structure consists of two identical light chains, polypeptides of around 25 kilo molecular weight, and two identical heavy chains, larger polypeptides of molecular weight 50,000 or more. Like the antibody molecules they constitute, H and L chains are also called immunoglobulins. Each light chain is bound to heavy chains by a disulfide bond and by such non-covalent interactions as salt linkage, hydrogen bonds, and hydrophobic bonds to form a heterodimer. Similar non-covalent interactions and disulfide bridges link the two identical heavy and light chain combinations to each other to form the basic 4-chain antibody structure, a dimer of dimers. As we shall see, the exact number and precise positions of these interchain disulfide bonds differ among antibody classes and subclasses. The first 110 or so amino acids of the amino terminal region of a light or heavy chain varies greatly among antibodies of different specificity. These segments of highly variable sequences are called V regions, VL in light chains and VH in heavy. All of the differences in specificity displayed by different antibodies can be traced to differences in the amino acid sequences of V region. In fact, most of the differences among antibodies fall within area of V region called complementary determining regions. And in these CDRs on both light and heavy chains that constitute the antigen binding site of the antibody molecule, by contrast, within the same antibody class, far fewer differences are seen when one compares sequences throughout the rest of the molecule. The regions of relatively constant sequence beyond the variable regions have been dubbed C region, CL on the light chain, CH on the heavy chain. Antibodies are glycoproteins with few exceptions. The sites of attachment for carbohydrates are restricted to the constant region. We do not completely understand the role played by glycosylation of antibodies, but it probably increases the solubility of molecules. Inappropriate glycosylations or its absence affects the rate at which antibodies are cleared from the serum and decreases the efficiency of interaction between antibody and the complement system and between antibodies and Fc receptor. So, chemical and enzymatic methods reveal basic antibody structure. Our knowledge of basic antibody structure was derived from a variety of experimental observations. When the gamma globulin fraction of serum is separated into high and low molecular weight fractions, antibodies of around 1,50,000 molecular weight, designated as immunoglobulin G, are found in the low molecular weight fraction. In a key experiment, brief digestion of IgG with the enzyme papain produced three fragments, two of which were identical fractions and a third that was different. The two identical fragments each with a molecular weight 45,000 had a antigen binding affinity. So here you can see the disulfide bond between two heavy chains, and between heavy chains and light chain, and we have talked about this. So if you have any doubt, you can let me know. We have already covered this. We'll now discuss about obstacles to antibody sequencing. So we will talk about obstacles to antibody sequencing. Initial attempts to determine the amino acid sequence of the heavy and light chains of antibody were hindered because insufficient amount of homogeneous protein were available. Although the basic structure and chemical properties of different antibodies are similar, their antigen binding specificities and therefore their exact amino acid sequences are very different. The populations of antibodies in this serum gamma globulin fraction consist of a heterogeneous spectrum of antibodies. Even if immunization is done with a hapten carrier conjugate, the antibodies formed just to the hapten alone are heterogeneous. They recognize different epitopes of the hapten and have different binding affinities. This heterogeneity of serum antibodies made them unsuitable for sequencing studies. Pure immunoglobulin obtained from multiple myeloma patients made sequencing possible. Sequencing analysis finally became feasible with the discovery of multiple myeloma, a cancer of antibody producing plasma cells. The plasma cells in a normal individual are end stage cells that secrete a single molecular species of antibody for a limited period of time and then die. In contrast, a clone of plasma cells is in an individual with multiple myeloma has escaped normal controls on their lifespan, and proliferations are not end stage cells, rather they divide over and over in an upregulated way without requiring any activation by antigen to induce proliferation. Although such a cancerous plasma cell called a myeloma cells has been transformed, its protein synthesizing machinery and secretory functions are not altered. Thus, the cell continues to secrete molecular homogeneous antibody. This antibody is indistinguishable from normal antibody molecules but is called myeloma protein to denote its source. In a patient afflicted with multiple myeloma, myeloma protein can account for 95% of the serum immunoglobulin. In most patients, the myeloma cells also secrete excessive amounts of light chains. These excess light chains were first discovered in the urine of myeloma patients and were named Bence-Jones proteins for their discoverer. Multiple myeloma also occurs in the animals. In mice, it can arise spontaneously as it does in humans, or conditions favoring myeloma induction can be created by injecting mineral oil into the peritoneal cavity. The clones of malignant plasma cells that develop are called plasmacytomas, and many of these are designated as MOPCs, denoting the mineral oil induction of plasmacytoma cells. A large number of mouse MOPC lines secreting different immunoglobulins classes are presently carried by the American type culture collections and non-profit repository of cell lines commonly used in research. Light chain sequencing revealed that immunoglobulins have constant and variable regions. When the amino acid sequences of several Bence-Jones proteins from different individuals were compared, a striking pattern emerged. The amino terminal half of the chain, consisting of 100 to 110 amino acids, was found to vary among different Bence-Jones proteins. This region was called variable region. The carboxyl terminal half of the molecule, called the constant region, had two basic amino acid sequences. This led to the recognition that there were two light chain types, kappa and lambda. In humans, 60% of the light chains are kappa and 40% are lambda. Now, whereas in mice, 95% of the light chains are kappa and only 5% are lambda. A single antibody molecule contains only one light chain type, either kappa or lambda, never both. The amino acid sequences of lambda light chains show minor differences that are used to classify lambda light chains into subtypes. In mice, there are three subtypes: lambda 1, lambda 2, lambda 3. In humans, there are four subtypes. So, heavy chain sequencing revealed five basic varieties of heavy chains. For heavy chain sequencing studies, myeloma proteins were reduced with mercaptoethanol and alkylated, and the heavy chains were separated by gel filtration in a denaturing solvent. When the amino acid sequences of several myeloma protein heavy chains were compared, a pattern similar to that of light chains emerged. The amino terminal part of the chain, consisting of 100 to 110 amino acids, showed great sequence variation among myeloma heavy chains and was therefore called variable region. The remaining part of the protein revealed five basic sequence patterns corresponding to five different heavy chain constant regions. Each of these five different heavy chains is called isotype. The length of the constant region is approximately 330 amino acids of delta, gamma, and lambda, and 440 amino acids of mu and epsilon. The heavy chains of the given antibody molecule determine the class of that antibody: IgM mu, IgG gamma, IgA alpha, IgD delta, and IgA epsilon. Each class can have either kappa or lambda light chains. A single antibody molecule has two identical heavy chains and two identical light chains. So, minor differences in the amino acid sequence of the alpha and gamma heavy chains led to the further classification of the heavy chains into subtypes that determine the subclass of antibody molecules they constitute. In humans, there are two subtypes of alpha heavy chains, alpha and alpha 2, and four subtypes of gamma heavy chains. Immunoglobulin fine structure. The structure of the immunoglobulin molecule is determined by the primary, secondary, tertiary, and quaternary organization of the protein. The primary structure, the amino acid sequence, accounts for the variable and constant regions of the heavy and light chains. The secondary structure is formed by folding of extended polypeptide chain back and forth. So class IgG, heavy chain, subclass light chain, molecular formula, and so on. So the chains that then folded into a tertiary structure of compact globular domains, which are connected to neighboring domains by continuations of the polypeptide chain that lie outside the beta pleated sheets. Finally, the globular domains of adjacent heavy and light polypeptide chains interact in the quaternary structure, forming functional domains that enable the molecule to specifically bind antigen and at the same time perform a number of biological effector function. And also, do you know that immunoglobulins possess multiple domains based on the immunoglobulin fold? We have covered this. We'll talk about the immunoglobulin superfamily. If you have any doubt, we can discuss. So, the immunoglobulin superfamily. The structures of the various immunoglobulin heavy and light chains described earlier share several features suggesting that they have a common evolutionary ancestry. In particular, all heavy and light chains classes have the immunoglobulin fold domain structure. The presence of this characteristic structure in all immunoglobulin heavy and light chains suggests that the genes encoding them arose from a common primordial gene encoding a polypeptide of around 110 amino acids. Gene duplications and later divergence could then have generated the various heavy and light chain genes. Large number of membrane proteins have been shown to possess one or more regions homologous to an immunoglobulin domain. Each of these membrane proteins is classified as a member of immunoglobulin superfamily. The term superfamily is used to denote proteins whose corresponding genes derived from a common primordial gene encoding the basic domain structure. These genes have evolved independently and do not share genetic linkage or function. The following proteins, in addition to the immunoglobulins themselves, are representative members of the immunoglobulin superfamily. IgG alpha and beta heterodimer part of the B cell receptor, poly IgG receptor, which contributes the secretory component to secretory IgA and IgM. T cell receptor, T cell accessory proteins including CD2, CD4, CD8, CD28. Class 1 and class 2 MSC molecules, beta 2 microglobulin, and invariant protein associated with class 1 MSC molecule. Various cell addition molecules including VCAM1, ICAM1, ICAM2. Platelet derived growth factors. Numerous other proteins, some of them discussed in other chapters, also belong to the immunoglobulin superfamily. X-ray crystallographic analysis has not been accomplished for all members of the immunoglobulin superfamily. Nevertheless, the primary amino acid sequence of this protein suggests that they all contain the typical immunoglobulin fold domain. Specifically, all members of the immunoglobulin superfamily contain at least one or more stretches of about 110 amino acids capable of arrangement into pleated sheets of antiparallel beta sheets, usually with an invariant intra-chain diacellate bond that closes a loop spanning 50 to 70 residues. You can see here. So these are some members of the immunoglobulin superfamily, a group of structurally related, usually membrane-bound glycoprotein. In all cases shown here except beta-2 microglobulin, the carboxyl terminal end of the molecule anchored in the protein. So monoclonal antibodies, as noted in chapter 3, most antigens offer multiple epitopes and therefore induce proliferation and differentiation of a variety of B cell clones, each derived from a B cell that recognizes a particular epitope. The resulting serum antibodies are heterogeneous, comprising a mixture of antibodies, each specific for one epitope. Such a polyclonal antibody response facilitates the localization, phagocytosis, and complement mediated lysis of antigen. It thus has clear advantage for organisms in vivo. Unfortunately, the antibody heterogeneity in that increases immune protection in vivo often reduces the efficacy of an antiserum for various in vitro uses. For most research, diagnostic, and therapeutic purposes, monoclonal antibodies derived from a single clone and thus specific for a single epitope are preferable. Direct biochemical purification of a monoclonal antibody from a polyclonal antibody preparation is not feasible. In 1975, George Kohler and Kaiser Mainstein devised a method for preparing monoclonal antibody, which quickly became one of the immunology's key technologies. By fusing a normal activating antibody producing B cell with a myeloma cell, they were able to generate a hybrid cells called hybridoma that possessed the immortal growth properties of the myeloma cells and secreted the antibody produced by the B cell. These are antigen with different epitopes, isolate spleen cells. So the plasma cells, the myeloma cells, hybridized hybridomas, select. So isolate serum, antibody 1, 2, 3, 4. Monoclonal antibodies have important clinical uses. So this is the now important part, the organization and expression of immunoglobulin genes.